Good morning, everyone. My name is Marge Poma. I'm a rising senior from Smith College, and today I will be speaking to you about my 10-week project in the Vitamin Uke lab on the transcriptional regulation of the crosstalk between iron and copper homeostasis. As plant scientists, I think we can agree that one of the biggest obstacles in agriculture is feeding the rapidly growing and malnourished population. Statistics show that nearly 1 in 9 people around the world are undernourished, and this population is expected to increase by 35% by 2050. The reasonable solution is to increase crop yield and nutritional value, along with using sustainable practices in the face of accelerating climate change. To achieve this, we need to think about micronutrients that are important for both humans and plants alike, and among these nutrients are copper and iron. So copper and iron are essential minerals for plant growth because they can easily switch between different oxidative forms, and this allows them to carry electrons to different parts of the cell and participate in electron transfer and any reduction oxidation reactions. So anything like photosynthesis and iron superoxide dismutases is where they would be needed. So plants are dependent on the availability of copper and iron in the soil. Something to note is that copper and iron are somewhat dependent on soil pH. So this is a map that shows different soil pH distributions around the world. And areas marked in blue and yellow represent alkaline and neutral soils respectively. And when the pH is equal to or above 7, iron and copper become less available for absorption by plants. Additionally, areas marked in red represent acidic soils that lead to toxic levels of iron. So toxic levels are represented in around 50% of ag soils, and iron and copper deficiency develops in around 30% of ag soils. So plants need to thrive in slightly acidic conditions in order to avoid this deficiency and toxicity. But how are plants surviving in extreme copper and iron deficiency and accumulation? So copper and iron deficiency is actually very difficult for plants to survive in. Here you have an example of low iron and low copper, and you can see that they reduce crop yield along with impairing photosynthesis and reducing pollen fertility. On the right, you can see that an accumulation of copper and iron can also kill the plant, as shown with leaf fluorosis in both pictures. So how are plants surviving in extreme copper and iron deficiency regulation? This means that they use regulation of expression of transporters in the roots. This figure represents a simplified version of the molecular pathways that maintain copper and iron homeostasis. In copper deficient conditions, SPL7 and CYF1 transcription factors part of the BHLH family are induced by copper deficiency and activate transcription of COP-T2 and FRO45 that work to uptake copper from the soil. Additionally, we have a dozen BHLH transcription factors that are induced by iron deficiency and work to activate IRT1 and FRO2, which uptake iron from the soil. And something interesting to note here is this arrow right here that tells us that BHLH transcription factors are also regulating COP-T2 and FRO45. And when one or more components of one signal transduction pathway affects another, we call that crosstalk. And so that crosstalk is what explains this graph here, where copper is accumulated under iron deficiency. And we believe this is a useful strategy for plants to use the oxidative potential of one micronutrient when the other is not present. We can see the opposite is also true with iron accumulation under copper deficiency. And we're not actually sure as to how this mechanism goes out, but we assume if one crosstalk is at play, then another crosstalk may be happening between CYF1 and possibly one of the BHLH transcription factors that are involved in iron deficiency. So CYF1 and BHLH transcription factors that regulate iron uptake may interact with each other on the protein level. And we have further reason to believe that CYF1 and these transcription factors may interact with each other because they are part of the basic helix loop helix family. Members of this family can form homodimers, as shown on the left here, but different BHLH members can form heterodimers. Therefore, it's very likely that CYF1 and another BHLH transcription factor can have protein level interactions. But how can we confirm this? Well, a used-to hybrid assay checks for interactions between two proteins, In this use2 hybrid assay performed by my mentor, Rujen, she identified the CYP1 interacting protein, which we call CITP, and we use CITP as the prey and CYP1 as the bait. We can see that our yeast was able to grow in 3 different mediums along with our control, shown here. This is our positive control, and this is our experimental. So yeast2 hybrid assay shows CYP1 and CITP protein interactions in vivo. Additionally, co-immunoprecipitation can also tell us whether 2 proteins interact or not using antibodies. My mentor also conducted this method using these expression cassettes used, and both were transformed into Arabidopsis thaliana using agrobacterium transformation. It's important to remember that anti-MYC is the antibody for CYP1 and anti-GFP is the antibody for CITP. This band here represents CITP, and because my mentor used anti-MYC for immunoprecipitation, this tells us that the 2 proteins are interacting with each other in planta. So now we go to our question, which is what is the function of the CYP1 and CITP complex? Going back to this figure, we hypothesized that this complex may be needed to uptake iron under copper deficiency, a phenomenon we have yet to understand. But before we answer this question, we also need to know in what conditions we can induce CYP1 and CITP protein complex to form so that it is the most stable. And to do this, we can look at the expression of CYP1 and CITP independently. So when we tested the expression and protein stability of CYP1 under copper deficiency, we found that the CYP1 transcripts were upregulated almost 30-fold. And we can also see that no CYP1 is expressed in iron deficiency. This is very important for our hypothesis because under normal iron deficiency response, CITP is upregulated to tell our transporters to uptake minerals. If CYP1 is upregulated as well, and if our hypothesis was true, then CYP1 could steal CITP from doing its job and form the complex. And so we think that the plants are destroying CYP1 to be able to allow CITP to undergo normal iron deficiency response. We can also see that protein stability was increased under copper deficiency using the expression cassette used above. And here we can see that there is more protein accumulated under copper deficiency with anti-actin as our housekeeping gene. So under copper deficiency, CYP1 transcripts were upregulated and protein stability was increased. When we tested the expression and protein stability of CITP under copper deficiency, we found that the CITP transcripts were not upregulated in copper deficiency, but that there was increased CITP protein accumulation in copper deficiency that in part depends on the CYP1 protein. We can tell because in figure B, there is more CITP available in wild-type plants under copper deficiency than there are in control. And we can also see reduced CITP protein accumulation in the CYP1 mutant, which is how we know that CITP is dependent on CYP1. Because our preliminary data show that CYP1 and CITP independently stabilize best in copper deficiency conditions, then we can assume that the complex can also stabilize best in these same conditions. So when my mentor used co-immunoprecipitation to detect both CYP1 and CITP using the transgenic line of Arabidopsis, we saw that the complex was found to have increased when grown in copper deficiency compared to control in not just 1, but 2 independent experiments. And when the solution was eluted, there was more CYP1 and CITP available in copper deficiency than in control. So under copper deficiency, CYP1 and the CITP protein complex accumulates. So now that we know the complex is formed under copper deficiency, our initial question remains, what is the function of that complex? One strategy is to observe the phenotypes of the CYP1 CITP double mutant. And just from looking at the phenotype of the CYP1 CITP double mutant from plants grown for 2 months shown on the left here, you can see the plants are very sick and not viable. So from the 3 and 4 plants that we see, we can tell that it closely resembles the CITP mutant in control conditions, which tells us that the CITP gene is upstream of CYP1. The next strategy is to observe the phenotypes of the CITP mutant under copper deficiency. And purely from observation, my mentor and I saw little to no changes in the phenotype of the CITP mutant under copper deficiency. This roundness you're seeing is something that's more of a copper deficient phenotype rather than CITP related. But it is possible that we're not seeing something. So as part of our next steps, we hope to use ImageJ to measure the size of the rosettes and conduct a qPCR analysis to see if the transporters in the major iron uptake system, RT1 and FER2, are expressed. Our next attempt is to observe the phenotypes of the CYF1 mutant under copper deficiency, which will hopefully tell us if CYF1 impacts any of the iron homeostasis molecular processes. We begin by observing the phenotypes of the CYF1 mutant under copper deficiency, and you can see that CYF1 does not have as strong of a phenotype as the CITP mutant did in the previous slide, but it manages to do just fine in our control. When harvesting the roots, we realize that there is a significant decrease in iron accumulation when the CYF1 gene is absent than when grown under copper deficiency, but when the CYF1 gene is present, there is more iron accumulated compared to control conditions. So if CYF1 is required for iron accumulation, does CYF1 also regulate the major iron uptake system, RT and FER2? To answer that question, we use qPCR analysis to test the expressions of the major iron uptake system, RT1 and FER2, and we see they are not affected in the CYF1 mutant, but that the overexpression line shows in these asterisks shows a significant reduction in the expressions of the iron uptake system. This is possibly to prevent iron from accumulating in the roots to prevent iron toxicity. Overall, we see that the CYF1 dependent iron uptake under copper deficiency is not through the major iron uptake system. And if it's not through the system, maybe there is another iron uptake transporter at play here. So another iron transporter may be involved in iron uptake from the roots under copper deficiency. Now that we know the major iron uptake system is not affected by CYF1 under copper deficiency, our next strategy is to use chromatin immunoprecipitation, or CHIP, assay to identify the direct gene target of the CYF1 CITP complex, as this would hopefully tell us what genes are transcribed and proteins are produced that contribute to iron accumulation under copper deficiency. In sum, we found that under copper deficiency, CYF1 is important for iron uptake. CYF1 interacts with CITP and the stability of the complex is increased. The CYF1 dependent iron uptake is not through the major iron uptake system. RT1 and FER2 downregulation in the CYF1 overexpression line could be to prevent iron overaccumulation and toxicity. And lastly, CHIP sequencing and RNA sequencing will identify direct downstream targets of CYF1 CITP complex and any new iron uptake transporters contributing to iron accumulation. I would like to acknowledge the Vadim and Uke Lab, especially my mentor, Ru Jen, for teaching me all that I know today: hydroponics, RNA extraction, qPCR analysis, just to name a few. I would also like to give a quick shout out to our funders, NSF, USDA, and IFA, for making this possible. I would also like to thank Megan Truesdale, Olivia Olstein, George Yander for leading another successful BTR-RU program, and Adrian Powell and Jing Zhang for teaching me the basics of bioinformatics. Thank you so much for listening.